this is an example on the acid-base catalysis that I have just explained up here. I hope this makes it easier. So basically you have a nucleophile, and this nucleophile attacks an electrophile. But what is a nucleophile? A nucleophile is a type of molecule that attacks another molecule and donates its electrons to that molecule, okay? And an electrophile is the electron acceptor. So let's say this is the nucleophile. It donates these electrons to this electrophile. So nucleophile, electrophile, okay? So the reactant species in this case are this and this. What happens is that it attacks this compound and an intermediate forms. Now, as I said up there, when the intermediate forms, it needs to be stabilized, right? Acid-base catalysis is a way that we can stabilize the intermediates that are found in the active site of the enzyme. Water acts fast and stabilizes this intermediate, so the reaction rate doesn't actually increase. But in this case, since water doesn't act immediately and some of the intermediate is breaking down back to its component reactant, then what happens is that an alternative proton donor, in this case it's gonna be the amino acid residue that acts as an acid, is gonna act fast and stabilize these intermediates, which increases the reaction rate. So the second type of enzyme catalysis is gonna be covalent catalysis. So the first type was acid-base catalysis. Now the second type is gonna be covalent catalysis. So in covalent catalysis, what happens is that a substrate that is gonna be bound to the nucleophile that is found on the active site of the enzyme, and this nucleophile is gonna be an amino acid residue like serine, cysteine, and histidine. So what happens is that a transient bond or a transient covalent bond is gonna form between the substrate and this, and it's gonna form this intermediate right here. Actually, this bond is necessary because what happens is that it becomes stabilized. To make it easier, you can think about amino acid holding the hand of this substrate throughout the crowd and leading the way. You can think of it like that, okay? So now, how do I recognize nucleophiles or electrophiles? So for nucleophiles, if you see a negative sign like here, 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 or these electrons here, here, and not here, these are electrons, and which means that these nucleophiles have gained these electrons and now they can donate these electrons, okay? Now in electrophiles, if you see this positive sign, or if you see a phosphorus of a phosphate group, or if you see an H plus, then that means, the plus means that they have lost these electrons, which means that they are acceptors of new electrons. These mean that they have gained electrons, which means that they are capable of donating these electrons, and these mean that they have lost and they are capable of accepting new electrons. These are nucleophiles and these are electrophiles. So, serine proteases. If we want to break down the name of this enzyme, so it's a protease, it means that it breaks down peptide bonds, and it's a serine. So this enzyme breaks down peptide bonds that are related to the amino acid serine, okay? So, like trypsin, chymotrypsin, and elastase. So what's the difference between these enzymes? They are actually 40% identical, but the main thing that's common between them is the presence of this triad. These three enzymes have a triad that's formed of aspartate, serine, and histidine. Okay, now I'm going to explain the function of this triad right here with the example of the enzyme chymotrypsin. So I want to give you a little bit of a generalized information about chymotrypsin. It's an enzyme that is released during digestion, and especially for proteins. So I'm gonna explain like how this triad is gonna break down the protein that you're gonna eat. For example, let's say you've had a filling protein meal, so chymotrypsin is gonna be activated, and this is the complex that's gonna mediate the reaction. So the first step is that we have serine, we have histidine, and we have aspartate. So serine is supposed to be the nucleophile that's gonna attack the peptide bond that's gonna come, right? But if you look at serine right here, it's not a very strong nucleophile. And it needs to be a strong nucleophile so that it can attack the protein, right? So what happens is that histidine pulls this hydrogen, it pulls it towards it, okay? And at the same time, this aspartate, or this oxygen in aspartate, pulls this hydrogen from histidine towards it. So it can look like this. If you can compare them, this H is now here, and this H is now here. This pulling makes serine a stronger nucleophile, okay? And this one is pulled for stabilization, okay? So, now, when does this happen? When does H get pulled by histidine? A substrate binds, okay? Histidine initiates the reaction. So, for example, a protein is bound to the enzyme, and what happens is that histidine immediately pulls this hydrogen towards it. It makes serine a stronger nucleophile. Moving on. So, serine now is a strong nucleophile, right? And this is the peptide the protein that we're talking about. Now, serine now is able to attack the protein, and I just want to explain a bit of organic chemistry so that you can actually understand what I'm saying or understand the reaction. When serine attacks this double bond should be broken, because according to organic chemistry, C only has 4 valence electrons, and it can only share 4 bonds, right? So if it has a double bond right here, and I'm trying to make a bond right here, so this should go back to this reactant. So this bond should be broken, and it goes back to oxygen in this form, and a bond forms here. So it becomes like this. Okay, so serine attacks, and it breaks this double bond, and it forms a tetramer that you can see right here. So serine formed a bond with the protein, and a tetramer broke, and these are the electrons from the double bond. Now, what happens is that histidine donates this hydrogen to this group right here so that it can release this amine group. And now after it donates it, as you can see right here, this amine group is released, and now we have a serine acyl intermediate. But we need the serine to come back to where it belongs, so we continue. Okay, now we need to return serine back to its family, right? So what happens is that aspartate and histidine activate water, okay? And water, what does it do? It donates, or it attacks this serine acyl residue, and a tetrahedral forms again, right? Now, since we want to release this away from the family right here, right? What happens is that oxygen returns its electrons back, and the double bond forms again. So it goes back to looking like this: C double bond O, R2, and OH. 1, 2, 3, and 4. Makes sense. So this is released, and serine goes back to its family. And this is called serine deacylation. Now you're gonna ask me, why did we do all of this if serine is gonna go back to how it looks? Because this reaction is breaking down or cleaving peptide bonds, and that's exactly what we did. If you can come back here, this is a protein that has been broken down by serine, right? Even though serine was bound to it and then this donated this to break it down, but that is the main purpose, right? Now, moving on to metal ion catalysis. This is the last type of catalysis, I think. So, metal ions act as electrophiles. How do they do that? They bind the substrate. By binding to the substrate, they orient it in the right position that it needs to be for the reaction to take place, and they stabilize it by accepting electrons. So the first example that was found in your lectures was about carbonic anhydrase, which is the enzyme that we're talking about, and the metal ion, which is zinc. So zinc that is found on the active site of the enzyme activates the separation of water into hydroxyl and H+. At the same time, carbon dioxide binds to the active site. So at the same time that water is separating into OH- and H+, CO2 is binding to the active site right here. And what happens is that OH- attacks the nucleophile minus, attacks carbon dioxide. And what happens? What forms? Bicarbonate. Okay? So bicarbonate is formed, it's released, and the cycle is repeated. Another example is the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is also related to cellular respiration, just like hexokinase that I mentioned in the beginning of this lecture. You're going to take cellular respiration, I think, in year 2 or year 1. I don't remember, to be honest. But pyruvate complex is made up of 3 enzymes. It's very complex. The first one is pyruvate dehydrogenase, the second one is DIT, and the third one is DID. These are the coenzymes of these enzymes, and what happens is that this is how they look like. This is enzyme 1, this is enzyme 2, and this is enzyme 3. In enzyme 2, there's something actually pretty cool. This is the arm of enzyme 2. It's like something that moves on enzyme 2, and it moves from enzyme 1 to enzyme 2 to enzyme 3. Okay, it's like a triad that moves, right? From active site E1 to active site to E2 and E3. Now you can ask me, like, why is there an arm that connects these 3 enzymes? That's because the connection between these enzymes doesn't allow the intermediates within these enzymes to go into solution, right? To dissociate into solution. I'll give you an example. So let's say pyruvate binds to this enzyme, and it binds on the active site, and it becomes acetyl, and carbon dioxide is released. And then what happens is that enzyme 2, through its arm, it binds to this acetyl, right? And it takes it to enzyme 2. In enzyme 2, it's bound to CoA. This is another thing related to cellular respiration. And then enzyme 2 then grabs the electrons from the compound that is acetyl CoA and donates them to enzyme 3, where NADH can be reduced. Now, I will not ask you to memorize this reaction in detail because this is not the aim of this lesson. I want you to understand the concept of this enzyme, of how the arm works. But if your professor told you or emphasized on this reaction in your lectures, then you should. But for me, this is just an example that helps you understand what is happening here. So, moving to inhibition of enzymes. This is actually pretty easy. So, what are the 2 types of inhibition? It might be reversible, it might be irreversible. Irreversible meaning that the enzyme is bound to something covalently. And when it's covalently, the bond is very strong. Even when you remove the thing that's inhibiting the enzyme won't work again. Okay? Activity won't be restored in irreversible, but in reversible it can be restored. A bond is gonna be a weak bond, not like the covalent bond. It's gonna be weak, just like hydrogen bonds. For example, is competitive inhibitors. So let's say you have an enzyme. This is the active site of an enzyme, and you have the substrate and the competitive inhibitor. It runs faster than the substrate and binds to the active site of the enzyme and beats it to the active site and inhibits the enzyme. That's exactly how a competitive inhibitor works. I'll actually link a video to a funny TikTok that explains competitive inhibition. And so what would happen to the reaction rate, the Km, Michaelis constant, and the Vmax in the case of competitive inhibition? So in this case, the enzyme is the same, right? Nothing is changing in the enzyme, right? So Vmax is going to stay the same because E is the same. But instead of having just the substrate, we have something other than the substrate, the inhibitor. But Km doesn't differentiate between substrate and inhibition. It just reads it as both are the substrate. For Km, the substrate is increasing. So what does that mean? Km is going to increase. Now, examples of competitive inhibitors. So this is not pharmacology. You're going to take these in pharmacology, but if your professor also emphasized on these in class, then you should know them. So an easy way to memorize these is, for example, all of these end with 8. So all of the ones that end with 8 are related to succinate. Methanol is related to alcohol, all. You have everything that ends with pril is a antihypertensive drug, so on and so forth. But if your professor didn't emphasize on these, I wouldn't actually memorize all of them. I would memorize maybe 2, 3, or 4 of them, and that's it. So your patient comes in with methanol poisoning. What do you do? So ethanol is a competitive inhibitor of methanol, okay, for the enzyme alcohol dehydrogenase. So we have an enzyme called alcohol dehydrogenase, and we have ethanol and methanol, right? So what happens is that methanol reacts with the enzyme and forms something called formaldehyde. Formaldehyde is very bad because it causes damage to the tissues and sometimes blindness. So what you can do as a doctor is that you put slow intravenous infusions of ethanol for several hours so that this slows the formation of formaldehyde, thus decreasing the risk of damaging the tissues. So what are transition state inhibitors? From its name, they are inhibitors that inhibit the enzyme at the transition state. An example of that is the inhibition of transpeptidase. Now, what is transpeptidase? Transpeptidase is an enzyme that synthesizes the bacterial cell wall called the peptidoglycan. Okay? So we as doctors, we prescribe or we give penicillin, which is an antibiotic that is a beta-lactam. A beta-lactam is just a ring that looks like this. It's called a beta-lactam. So what happens is that penicillin goes and it inhibits transpeptidase. And when you inhibit transpeptidase, you don't synthesize cell wall. When you don't synthesize cell wall, you have no protection and the bacteria is dead. And anytime you see in my notes when I have this written, this means it's inhibition. This means it's inhibition. The second example is HIV. So HIV is a retrovirus. Retro from Greek means return or backwards, right? So what that means is that HIV has an enzyme called reverse transcriptase. Reverse transcriptase has the ability to change RNA to DNA. So HIV has RNA. Using this enzyme, it can make its RNA DNA. After that, it takes this DNA and injects it into our cells, the host cells, right? So what happens is 2 scenarios. The first scenario is that this DNA injected into us remains dormant for very long. Or it immediately gets transcribed and translated into proteins. These proteins are arranged like skyscrapers in our cells. And the HIV, the virus actually, has an enzyme called the HIV protease that breaks down these skyscrapers into the virus particles and synthesizes new HIV viruses. So, us as doctors, we target this HIV protease and we give drugs that inhibit it, of course, at the transition state. So, moving on to the uncompetitive inhibition, the exact opposite of competitive inhibition. The inhibitor looks nothing like the substrate. It will not bind to the active site and it will not compete with the substrate. It will bind to its own side and it will deactivate the enzyme. The KM and VM make sense because we have deactivated enzyme. Now, mixed inhibition is when the inhibitor binds to its own site, not the active site, or binds to the substrate enzyme complex, but not on the active site. Okay? So these are examples of irreversible inhibition agents. Personally, I did a mnemonic back then and I studied the mnemonic. So if you like mnemonics, then do memorize it. If you don't, then that's up to you. So DID, did. Heavy metals is he. Chelating is cheat. And phosphofluoridate is Phoebe. So if you know Friends, the show, then you would know who Phoebe is. And again, if your professor didn't really emphasize on these, then I wouldn't really focus on them. But if she did, then you should. The last one is mechanism-based inactivators cause suicide. So these inhibitors, they look exactly like the substrate. They bind to the active site right here. They bind to this active site and they undergo metabolism just like a normal substrate. But at some point in the metabolism, they form an intermediate that irreversibly binds to the enzyme and deactivates it. When it deactivates this enzyme, the cell becomes confused because it cannot perform cellular respiration, it cannot breathe, okay? And thus the cell kills itself. I'll give an example. Oxaloacetate is a metabolite related to the Krebs cycle. The Krebs cycle, again, is related to cellular respiration. And cellular respiration, as I said before, generates ATP. So if the cell cannot respire or generate ATP, then of course it's going to die, right? So what happens is that oxaloacetate binds to CoA, and this forms something called citrate. Citrate then activates an enzyme called aconitase, and this activates cellular respiration. Fluoroacetate looks exactly like oxaloacetate. When it binds to CoA, it forms something called fluoroacetyl-CoA. When fluoroacetyl-CoA binds to oxaloacetate, it forms something called fluorocitrate. Fluorocitrate inhibits aconitase. Remember, we need aconitase to activate cellular respiration. If aconitase is not activated, the cell cannot breathe. If the cell cannot breathe, the cell will kill itself. This is called mechanism-based inactivation, which is the coolest one in my opinion. Another example of suicide inactivators is the allopurinol drug. So you're a doctor and you suspect your patient has increased levels of uric acid. You give them allopurinol drug, and how this happens is that in our bodies there's an enzyme called xanthine oxidase which catalyzes the formation of uric acid. It increases the levels of uric acid. So when xanthine oxidase binds to allopurinol drug after you prescribed it to your patient, something called alloxanthine forms. Alloxanthine, when it forms, uric acid will not be formed, right? So when uric acid won't be formed, then your patient won't have a high risk of having crystallization in their kidneys because uric acid forms kidney stones, and when it accumulates in the joints, it may cause gout. Or this is like a uric acid related type of arthritis. This also emphasizes this example, and this example emphasizes the specificity of suicide inactivators, because if fluoroacetate didn't look exactly like oxaloacetate, it wouldn't have been able to deactivate aconitase, and the same thing applies for allopurinol. So now moving on to enzyme regulation. If you think with me right now, reactions that are at equilibrium, do you think these reactions need regulation? Of course not. They're already at equilibrium. What type of reactions that need regulations are those that are very far from equilibrium, right? How? Actually, we use enzymes to regulate these reactions. So, you regulate the enzyme that is responsible for the rate limiting step. What is the rate limiting step? If I have to reiterate, the rate limiting step is the slowest step in the reaction, and this reaction actually tells us the flow of the reaction. If it's going to be fast, if it's going to be slow, it literally tells us what the speed of the reaction is going to be. The rate limiting step tells us the speed of the reaction. It's the slowest step in the reaction, and most oftenly it's the first enzyme of the reaction. I gave you an example of what a rate limiting step might be in real life. So let's say these cars are moving fairly well, and then this car blocks the way of the other cars. So think of this as the rate limiting step. It slowed down the movement of the cars behind it, and that's exactly how you might think of a rate limiting step, just to make it easier for you. So how does the cell regulate these enzymes that are related to the regulation of reactions? It either increases or decreases the production of this enzyme. How? Through, number one is allosteric modulation of this enzyme. Allosteric modulation means that I change the shape of this enzyme. It looks like this, I make it look like this. Reversible covalent modification, something binds to this enzyme and it changes the activity of this enzyme. It's either activated or deactivated. Irreversible covalent modification, it's more of a permanent change to the enzyme. And then the last one is the binding of regulatory proteins that either activate or deactivate this enzyme. Now, what are allosteric enzymes? Allosteric enzymes are enzymes that are controlled by allosteric modulators, right? So they are enzymes controlled by modulators. They have different sites for different modulators. If you look right here, its shape, it's a complex machine with different parts. This is the active site, this is the regulatory site, the performance. So these modulators that the enzyme binds to affect the sensitivity of this enzyme. It's either highly sensitive to the substrate, which means that this enzyme has high affinity to the substrate, or it has low affinity to the substrate. Okay? And then what is the benefit? The benefit of having allosteric enzymes within a cell is fine-tuning. The cell would be able to fine-tune its own chemical reactions. The cell will have more control over the reactions that are happening. For example, within the moment, the cell decides that I need to speed up this reaction. So it uses allosteric modulators to modify enzymes immediately. Okay, that's more of a fine-tuned type of reaction or control. Think about it in a way of having a blurry image and then putting on your glasses, and you're gonna have perfect vision. So lastly, what are the types of allosteric enzymes? We have homotropic and heterotropic. Homotropic is when the substrate itself is a regulator. Like on the active side, when the substrate binds here, it regulates the activity of the cell. And heterotropic is when the modulator other than the substrate needs to control the enzyme. So we have on the regulatory site, we have a moderator that binds here, and then we have a substrate that binds here. That is different. Why don't allosteric enzymes follow the Michaelis-Menten kinetics? Because in allosteric modulation, what happens is that when a modulator binds to the enzyme, it changes how this enzyme is gonna react to the second substrate. I'll reiterate. In Michaelis-Menten mechanics, an enzyme normally binds to a substrate, it forms a product, and that's done, and then another substrate that looks exactly like this one binds, and then the product forms. This is exactly very simple, very easy. And we get this curve if you remember. We have Km, we have Vmax, whatever. But in allosteric enzymes, what exactly happens is that let's say there's two sites, okay, and the substrate is bound here and the modulator is bound here. The modulator is actually going to change the enzyme, and when let's say the enzyme has changed, for example, this is going to affect how the second substrate is going to bind to the enzyme, right? So the curve is not going to look like this one. This is a hyperbole. This is a hyperbole. But this one is more of a sigmoidal looking curve. So to reiterate, since this one is not like this one, because this one, the shape of the enzyme is changing, or the activity of the enzyme is changing, and when the activity of the enzyme or the shape of the enzyme is changing, this means its interaction with different substrates is gonna be different. However, this one, enzyme is not really changing. It binds to the substrates the same way, whether it be the first substrate or the 100th substrate. So this is an example. This enzyme that's called aspartate transcarbomyolase. So this enzyme exists in an inactive and an active state based on the presence or absence of ATP and CTP. So the exact function of this enzyme is building pyrimidines. What are pyrimidines? Pyrimidines are the base pairs that are the building blocks of DNA and RNA, such as cytosine, thymine, uracil. Okay? So when this enzyme sees that ATP is binding to its regulatory site, it recognizes that the cell is not metabolically active, which means that the cell does not need base pairs to build new DNA. So it inactivates itself. But when CTP is not bound, which means that there is a high ATP in the cell, the enzyme recognizes that the cell is metabolically active. The cell is metabolically active, which means that this enzyme gets activated and it builds pyrimidines for DNA and RNA. Now, mercury is a toxin that desensitizes this enzyme, and of course, when it's desensitized, it won't be able to build pyrimidines properly according to the cell's needs. Now, this is just a reiteration of what I have previously explained before about the modulators of enzymes. So let's say a cell receives a signal, for example, in the form of a hormone, and this causes genetic change that leads to transcription and translation of new proteins. And these proteins then go and regulate the enzyme. So this enzyme might be degraded. It might be sequestered into the ER, endoplasmic reticulum. It's not broken down. It's just taken back when it's needed again. So it's like putting it in the closet and I'll use it again when I need it. okay? The enzyme either binds to the substrate that's been synthesized. We have phosphatases and kinases. You phosphorylate or dephosphorylate the enzyme. We have the allosteric effector that we talked about, and we have the regulatory proteins that bind to enzymes. So, what is feedback inhibition? I'll give you an example of hormones that are found within our body. So, in our brain, we have the hypothalamus and the pituitary gland. The hypothalamus secretes something called thyroid releasing hormone. It goes to the pituitary gland and activates it, and the pituitary thus releases thyroid stimulating hormone. Thyroid stimulating hormone then goes to the thyroid gland, and the thyroid gland is activated to release the T3 and T4. So, what happens here is that T3 and T4, when they are released from the thyroid gland, they act as allosteric modulators. What did I say allosteric modulators were? They were substances or proteins that act on the enzyme and inhibit it, or change its shape, or deactivate this enzyme, right? So T3 and T4, they go to the hypothalamus and they deactivate the TRH. But in this case, TRH is not exactly inhibited. What happens is that there's just decreased production of TRH instead of inhibiting it, right? So if you want to think of it in an easier way, for example, let's say you have a company and you have a lot of workers. You don't just fire all of them, right? You just hire less workers. And that's exactly how T3 and T4 inhibit TRH. So if I want to reiterate what I just said, feedback inhibition is an allosteric modulator that inhibits an earlier enzyme in the pathway. So these enzymes inhibit the earlier enzyme that's coming from the brain. So explaining the multiple feedback loop and the simple feedback loop. So these are the enzymes S1, S2, S3, S4, and these are the products of these enzymes. Okay, so what is the multiple feedback loop? So let's say product B, okay, the multiple feedback loop is the inhibition of the enzymes that were not exactly prior to B, but early. So it's this one and this one. So it inhibits the earlier enzymes through the multiple feedback, which is the red line, and through the simple feedback loop, it inhibits the enzyme just prior to it, which is S4 in this case. Think of it like a city with intersections controlled by traffic lights, and these products are the traffic lights, okay? It's very easy.